Happy Thursday, Limburg Science students. Hopefully no one's too overwhelmed by this chapter so far. Sorry the video the other day was so much longer than normal. We're going to cut it down really as much as possible today. It should be pretty short. So let's jump into this right here. Where is DNA stored? We've been doing this since the beginning of the year, and hopefully you remember by now that DNA is in the nucleus. Nice and safe, nice and protected. We want it to stay there. How do the 4 DNA bases pair up? Again, hopefully from the other day you remember you've done a lot of practice in the last couple of days. I hope. You should know by now that A pairs with T, adenine with thymine, and C cytosine pairs with G guanine. That's the basis of this entire chapter, no pun intended basis. But yeah, that's what this really entire chapter is built on. A with T, C with G when you're talking about DNA. That little bit of information lets us do everything. So why is it important that bases are zipped up when they're not being used? So why do we want it that our DNA looks kind of like this with our letters sort of just in the middle? Why do we want it that our DNA is sort of like a ladder like that and not just kind of this? Why would it not really be great that if it kind of opened up like that? Well, it's much safer, right? If it's closed up like this over here, it's going to be protected. The bases are not exposed to the environment. They're a lot less likely to be damaged or some kind of horrible change happening to our DNA. Last question, we talked about one thing briefly the other day. What are 2 reasons that a cell may actually try to do this and unzip the DNA for some point in time, some period in time? Why would it want to open up that DNA occasionally? Well, the one thing we saw the other day is if you want to make a copy of it. If maybe you're going to make a new cell, you have to split your DNA and make 2 copies of it so that one can go to each of the new cells. Otherwise, somebody's not going to get enough. That's one reason to do it. The other one we haven't really talked about, but any ideas? Why would you sometimes want to be able to really to access these guys in the middle? Why would you maybe be wanting to go and be able to actually look at those letters to see are they A's and T's or C's or G's? What is DNA? DNA is just kind of the recipe, right? So why might you occasionally want to take a look at a recipe? That's going to be the next couple of days we'll take a look at. The big reason to do this is to actually use the recipe. If you actually have to follow the instruction manuals in DNA to make a protein at some point. That's going to be a pretty important process. We're going to get a very, very brief look at that today, the start of the process, and then next week we'll get much more in depth. We'll do a little bit of drawing, but today we're going to keep it short and sweet and take a look at the start of reading that recipe. So back in the beginning of the year, when we first talked about these big organic macromolecules, we moved very quickly through nucleic acids because I said we didn't need to know it then. We'd spend a lot of time later in the year. Now we're getting into that. The other day I gave you a little bit more focused explanation of DNA, the structure, what it looks like, how the bases pair up, how we know these things, the history, the discovery of the molecule, things like that. Today we're going to look at something we really haven't spent any amount of time talking about, I don't believe, and that's going to be RNA, the other white meat of nucleic acids. So we did DNA the other day. We also looked a little bit about how DNA copies itself, how it can unwind and just sort of build on the 2 sides of the molecule to make 2 identical copies of itself. So let's just refresh our memories before we get into anything new, and that's going to be a process we call transcription that we're looking at today. So we saw this picture the other day. If we have this green molecule of double-stranded DNA, but want to make another copy of it, so let's say we want to copy the cell, we need to copy the DNA first, how would that happen? The structure of DNA makes it really easy to unzip or open up the ladder or the double helix. All we have to do is basically just kind of cut, open it up right down in the middle here. We're going to open it wide up, and that'll help us make 2 new copies of what was already there. Get the last little bit of ink. All right. So doing this, opening it up, would expose the nitrogenous bases inside, all those A's, C's, G's, and T's, making it look like this, 2 single strands. I only actually show you one of them to save space here, but there would be another one. This is more of the left-hand strand here. This one would be somewhere else doing the exact same thing we're going to say. But this is now a single-stranded side, 1/2 of that original green DNA molecule. Now, opening up like this also allows the cell to then add brand new DNA bases to each of the single-sided strands. So where there's an A, another letter would come in here and be added to it. Where there's a C, another letter would come in there and be added to it, and so on, all the way down that new thing. This now forms 2 completely new double-stranded DNA molecules. Again, I only showed you one of them, with 1/2 older DNA, that's the side right here that's shown in green, and 1/2 newer DNA that was just added, that's the side shown in this kind of yellow orangey color. Now, based on what we saw the other day, what should these bases be? If we have an A, a C, a G, an A, and a T on the one side, what should the other side of the molecule look like? Hopefully from the stuff you did a lot of practice with the other day, or maybe you're still getting to it now, across from an A, we're always going to have a T. Across from a C, we're always going to have a G. Across from a G, we always have a C. Across from an A, we have another T, and across from T, we have another A. It's always going to look like that. And if you notice, this brand new double-stranded DNA molecule right here is an identical copy of the original double-stranded DNA molecule that we started with. And I only showed you one half here, but this side of the molecule that was green there will be somewhere else having other orangey bases added to it, and it'll also look identical to this molecule right here and to this molecule right here. So that's how the process of DNA replication works. Hopefully that makes sense from the other day. If not, once again, email me with any questions, come to the virtual extra help on Microsoft Teams, and we'll get you straightened out. We talked a lot about how DNA is the instructions for making proteins, kind of like a big recipe book. But does anyone remember who uses these instructions to actually make the proteins? Back from the beginning of the year, we did cell bio. That job belongs to the ribosome, those small organelles in the nucleus. Sometimes they're stuck to the endoplasmic reticulum, sometimes they're by themselves. They have this two-part shape in the top right here that I always think kind of looks like one of the Goombas from the original Mario games, but usually see them just drawn as small circles, because when we're looking at the entire cell, it's really kind of pointless to draw this more detailed shape. All you have to know is that there are these little things in the picture there at the bottom right. So does anyone see a problem here with how this whole process would work? The DNA instructions are in the nucleus, but they need to get to the ribosomes outside the nucleus in the cytoplasm. So how is that supposed to happen? If you think back to that diffusion lab we did a long time ago in the beginning of the year, remember with the bag and it changed colors on the inside but on the outside? Well, why did that happen? Why couldn't the starch cross the membrane to get out of the bag, but the iodine and glucose molecules could? Remember why that happened? Well, that's because the starch was a big molecule and couldn't fit through the membrane. It had tiny little microscopic holes that we can't see, but some molecules are able to pass back and forth while others couldn't. So do we think that DNA, a notoriously huge molecule, is going to have much better luck getting out of the nucleus to the cytoplasm? And would we want it to even be able to do that in the first place? Do we want it to be able to get out of the nucleus? Not so much really to either question. And that's going to be where RNA comes in. DNA is just too big to pass through the membrane surrounding the nucleus, and we don't really want it to leave anyway because it's safe in there. Instead, it makes a copy of the specific instructions it needs at any one point in time. So when a protein is needed, the section of DNA that codes for that protein opens up, it unzips. This is like turning to one page of a cookbook that has the recipe you want at that point. Instead of now a new copy of DNA being made, like we saw the other day, where an entirely new DNA molecule will be created, in this situation, the other nucleic acid, RNA, comes in and matches the exposed bases to make a short copy of the DNA recipe. So this process of copying DNA into RNA is called transcription. Transcribing means to make a copy. If you transcribe something, you would be copying one thing into another. And that's what's happening here. The DNA unzips so that a short RNA copy can be made, and then the RNA leaves so that the DNA can close back up again. Just what is RNA and how does transcription work? All we've talked about RNA so far is that it's a nucleic acid and is similar to DNA, which we do know a few things about at this point. DNA stands for deoxyribonucleic acid, so by the same kind of logic, RNA then is ribonucleic acid. It's very similar. It has the same kind of matching rules that we use that word complementary for that DNA does. So specific bases and letters of DNA always pair with each other, and the same is true for RNA. In this picture, we see a blue-colored double-stranded DNA molecule, which is then opened up in one area so that the orangey-colored RNA can move in to match up with the instructions that the cell needs. The short RNA sequence then separates and leaves the nucleus to deliver the instructions to a ribosome, which will read the recipe and make the desired protein. You can see here then the DNA closes back up again, goes back to normal, and the RNA leaves to go do its job, deliver the instructions somewhere else. Before we do a quick comparison between DNA and RNA, let me point out that there are actually two different types of RNA. It's not just one generic thing RNA. There are two types of this thing. This type that we've been looking at right here is called mRNA, which stands for messenger RNA, because that's what it is. It's a messenger that brings the message of instructions from the original DNA out to the ribosome. And the other type of RNA that we'll talk about next week is called tRNA, which stands for transfer RNA. That's because it transfers or moves something in a cell when a ribosome is ready to make the protein. So the mRNA is a copy of the recipe, and the tRNA is going to be different. It's going to be the thing that delivers the ingredients needed to actually follow that recipe and put the protein together. So whatever the ribosome needs, the tRNA, the transfer RNA, will deliver those materials to the ribosome so that it can actually do its job. And notice how just when we write these things, DNA is capitalized, all uppercase letters, RNA is capitalized, all uppercase letters, but we write the M or the T in messenger RNA, mRNA, and transfer RNA, tRNA, respectively, as lowercase letters. So the little lowercase m and lowercase t are just telling us which version of RNA we're talking about. So those get written lowercase, the other letters all uppercase. Just something to point out there. Let's compare our two nucleic acids, DNA and RNA. What's the first thing you probably notice that's different between these two if you look at the picture up top? As you can see here, DNA is a double-stranded molecule and RNA is single-stranded. So RNA is like one side of the DNA molecule, where we would say DNA is a double helix because there's the two sides twisted around each other. You could just say RNA is more like a single helix. As we've seen say, DNA is the original instructions or information or recipe, and that stays in the nucleus, while RNA is a copy that leaves the nucleus to deliver the instructions. I usually make the analogy of how you can go to the school library and get a textbook to copy or take a picture of, but you can't actually borrow those books that are behind the desk. DNA is the real book that stays put, while RNA is the copy that can be taken out to copy or take a picture of. RNA carries the recipe somewhere else because DNA is not allowed to leave to do it. So another distinction or difference is that DNA stands for deoxyribonucleic acid, as we've seen, because it's just the type of nucleic acid that has this sugar called deoxyribose up top at the left. So it's just, what does the name mean? RNA, the same way, stands for ribonucleic acid, as we saw a second ago, because it's the type of nucleic acid that has this sugar, ribose. And if you look at these two pictures up top, the only difference is that these highlighted parts right here, backtrack one sec, get my pen back, there we go. The only difference is that deoxyribose has an H, and ribose has no H. The deoxy part of deoxyribose just means without oxygen. So one molecule has an H, the other one has the OH. And I clicked the wrong thing, so it went ahead and added the next bit of notes right there. So let's see what that says at the bottom. Last but definitely not least, the biggest thing we need to know about these two molecules is that DNA has the four bases A, C, G, and T, which we've done a little bit of already in the last week. A always pairs with T, C always pairs with G. This is what the T, the thymine, actually looks like at the bottom. You don't need to know that structure, but I just want to show you for comparison. RNA, in contrast, has three of the same bases, A, C, and G, but instead of the T, the thymine, RNA instead has a different fourth base that we call uracil, or U. And this is what the U looks like. So again, just look at the picture. You don't need to know what these things look like. I'm never going to ask you which one is which, but just look how similar they are. For the T, it has a C and three H's up on the top left here, and the U only has an H. So they're very, very similar structures, just as the two sugars up top are very similar structures. This substitution of U for T in RNA compared to DNA is what you need to remember. So when the RNA copy is made from DNA, like we saw a minute ago, it'll always have a U where there was an A, instead of making a copy that's a T for where there was an A. C and G stay exactly the same. Those rules don't do anything different, but there's never going to be any T's in RNA. There's always going to be a U instead. So right here, this is the main thing we got to remember here. C and G work exactly the same, but U is different in RNA. Now, if you had a T in DNA, that would get copied to an A in RNA the same way. But if there was an A in DNA, it'll get copied to a U instead, not a T. So there's never going to be any T's in RNA. That's the important thing to remember. That's all the new stuff for today. I told you it's going to be short, so let's just do some practice stuff here. I know we're at almost 14 minutes already in this, but let's just get some practice stuff out of the way, and then I'll give some work to do. So we've got a Venn diagram here comparing DNA and RNA, and how are we supposed to know anything? I don't know if we have any clues here. The only thing different on each side is that the left side says it's a short molecule, and the right side says it's a long molecule. So if one's going to be shorter than the other, we know the short molecule is going to always be the RNA, and the bigger molecule, the much bigger molecule, is going to be the DNA. So that's what we're going to look at now. Now we can fill these sides in. Let's do DNA on the right, because we know a little bit more about that. We're a little more experienced with that. The bases are, I'm not going to write the whole name out. You can if you want to. A stands for adenine. T stands for thymine. C is cytosine. And G is guanine. And I'm actually going to write the T, because that's the important one that we want to remember is in DNA as a distinction. On RNA, and the other side, it still has A is adenine. I'm going to skip a line, and I'll put C and G still down there for cytosine and guanine in RNA. But now we know that instead of the thymine on the right, RNA instead has this other letter U, and that stands for uracil. So that's the one distinction we're going to make between DNA and RNA in terms of the bases. The sugar in RNA is going to be ribose, That's the R part of RNA. It's ribonucleic acid. And in DNA, the sugar is deoxyribose. For both of them in the middle, we only have 4 bases. It's just the 4th base is different each one. 3 are the same, and the 4th one is a little bit different. They're both chain-like molecules. They're both little pieces of repeating units. And the 3 parts of nucleic acids here are phosphate, sugar, and base. That should say nucleotides. I don't know why it says nucleodes, but it should say nucleotides up there. Okay. Just want to mention that Venn diagram from a second ago. I gave you that in your notes, but I'm pretty sure I also made the first page of your homework, so feel free to skip that page. Just write on it. We did in the notes, whatever. As long as you get the gist of it, that's the important thing. So let's look at a couple of questions here to finish up. One multiple choice question says, what is the complementary messenger RNA sequence for the DNA sequence shown below? So these 6 letters right here, CAAGGT, are going to be the DNA, and we want to figure out what the mRNA sequence that would match it when it makes that short copy would look like. So let's just go match our bases. That's the only thing to do. When I see a C, I'm going to write a G. When I see A's, now remember there's no T's in RNA, so in RNA, any A we match up by a U, so I'm going to write a U here and a U there. G, I'm going to write a C, another G, I write another C, and T still translates over to A, so it should look like GUUCCA, and right there, choice B is what that would look like. Now, considering we want to make it look like RNA, if you remember that RNA does not have T's, you can immediately cross off this choice because there's T's, and cross off this choice because there's T's. And then you're left with 2 choices only, and now narrow down your options. Even if you need to guess, a 50-50 shot of getting it right. But B is the correct answer there. Next one says, base your answer to the following question on this picture. And it says that the segment of DNA represented by the diagram was used as a template in the synthesis of messenger RNA. So I want to again make an mRNA sequence. Which sequence represents the order of bases found in that mRNA molecule? Now, they don't tell us which side we're copying. Are we copying this side here, or are we copying that side there? And that's going to make a difference. We don't really need to know, though. So let's just go through and we'll fill our letters in as we would. Let me erase this quick. If we're going to do the one on the left, I'm just going to sort of write over to the left here. A would become a U in mRNA. This A would also become a U. T would look like A on the mRNA. G would have a C matching it, and this C would have a G matching it. If instead we were copying the right side, T would then look like an A. T again is an A. A in DNA would make a U in RNA. C would make a G, and the G in the template would have a C in RNA. So we can just look for either one of these sequences because they didn't tell us which one we care about. And just looking at our answers down here, I see UUACG as my 4th choice. That's this one right here. So that looks like it's going to be the answer. Much like up top, we can eliminate choice C and eliminate choice B because they have T's. Actually, A also has a T in the middle there. Those 3 answers all have T's in them, and we know RNA should not have any T's. So right off the bat, we can eliminate 3 of our 4 choices, and now that gives us the right answer. Or if you have to go through it like I did, you could just do both choices, both options, both sides, and see which one matches one of the answers. So a little bit of stuff to check on, but it all works. These questions here from the second homework page I gave you today after the Venn diagram sheet. So before we do anything, let's just acknowledge that whoever originally made this page spelled complementary wrong. That is one spelling of it, but it's not the one we care about. And other than that, let's jump into here and just match some stuff up. And we got to carefully read these questions because the first question says write the complementary DNA strand. So this just says if the sequence given is DNA, what would the other half of the DNA look like? So we're going to keep the same letters A, T, C, and G and just do what we did the other day. Anywhere I see a C, I'm going to write a G, G becomes C, T becomes A, A's become T's, G becomes C, C would have a G across from it, G has a C across from it, C has a G, T is across from an A, A's are across from T's, T's are across from A's, and A's across from T. That's what that one looked like. You can do the next one by yourself because you've been doing it for a couple of days now. You're just matching up A's with T's, C's with G's. On the next set of questions here, now it says write the mRNA strand. So that's why it's underlined. It's important. Now, given this DNA sequence, you want to write the matching mRNA sequence that would be made as a copy to be sent out of the nucleus. So you're not just going A to T, G to C. This is where you have no T's. You're going to only write U's instead. So right away we have to do that because the first letter is an A. So if the DNA had A there, the mRNA copy would have a U across from it. T still goes to A, G still has C, T has A, I'm going to write that a G, a C, a G, A, C. Another A gives us a U, again an RNA. T still has an A becomes a U, C has G, T has A, G has C, and T has A. Same thing can do with the next question, and we'll move on. One last thing to show you here. I'm assuming this is probably easy to figure out, but I just want to make sure in case there's any questions about it. However this page was formatted just got weird at some point by whoever made this page. So these question marks in the middle should be either arrows or dashes or even some kind of equal sign. It's just showing that in RNA, going from DNA to RNA, an A becomes a U or is matched up with a U, T still goes with A, and G goes with C. So I don't know why it's a question mark there. It's just a different symbol, I guess, got inserted. But it's just showing you the base pairing in RNA compared to DNA. So if you're confused with that, just let me know. I'll go over anything you need to, but that's all that's doing. That's all for today. So go finish today's form. I think it's 6 questions, and then complete today's homework at some point. This should be posted on Thursday, but you have until Monday night to finish the work from this week. I know that Tuesday was a lot of intro stuff to set the groundwork. Hopefully everything today goes a lot faster now that you get more practice and more experience and kind of just used to filling these matching letters. I'm doing one more Microsoft Teams Extra Help Conference tomorrow. That's Friday the 17th at noon, and then another one on Monday the 20th, also at noon. So that's 2 chances to ask questions before the work is due, even if you don't even start until the end of the week. We did a lot this week, and I expect people to have questions. If it looks weird at this point, it's supposed to. It's a totally new set of material, stuff we really haven't done this year yet. So email me any questions, come to Extra Help. I'll get you on track for this chapter. Have a good weekend. Stay safe. Check in with each other and look out for each other, and I'll talk to you next week.